Okay, so first of all, I'd like to thank the organizers for inviting me. And as I was preparing for this conference, I was reminded of a conference that I've been here as a graduate student more than 20 years ago in Jerusalem. And it was a very inspiring conference because it was a stimulating time about new algorithms about learning. And today I feel that it's also a very exciting time in the field and the interface between neuroscience and deep learning. And what I hope today is to convey my enthusiasm to this, as well as maybe seed some new ideas. So my ultimate goal is to understand the brain computations that enable face perception. So the question in my mind is whether we can leverage advances both in neuroscience as well as in deep neural networks to produce a neurally accurate model of face perception. So in order to do such thing, we need to do a couple of things. First, we have to understand how the human brain works and understand implementation properties of the brain. Second of all, we need to implement these properties into hierarchical deep neural networks, or HDNNs. And then what we could do is use these HDNNs to test the computational utility of specific implementation features of the brain. And the reason that this is an exciting time in the field is because of work like the work by Dan Yammans and colleagues have shown that by taking a neurally inspired brain network and training it to do an object recognition task, a network like this can not only predict human performance, but it generates neurons or units in the network that resemble the responses of neurons in the macaque brain. And one of the reasons that so not only is this hierarchy, this neural network is inspired by the brain, but it also has several useful computational features. First of all, it can map any function between input and output. Second of all, it is trainable. And third of all, once it's trained, it's very fast because of the feed-forward processing. So what happens in the human brain? So imagine that you're looking at this face. Light gets reflected from the face to your eyes, and an image is formed on the retina. Then through the optic nerve, it reaches the LGN, and from the LGN, it reaches primary visual cortex, or V1. And then through a series of visual areas that we think of them as processing stages, information arrives to what I'm going to call as ventral temporal cortex, or VTC, and then maybe a percept emerges. Now, in the domain of face recognition, we might need to think about which regions might be specifically involved in face processing. And in the ventral streams, there are 3 clusters of face-selective areas in the human brain. And the way that we identify these regions in people's brain, we put them in an fMRI scanner and shows them a multitude of images, and then search for voxels in the brain that respond more strongly to faces compared to other stimuli, including body parts, common objects, places in building, as well as characters. And typically, you'll find 3 clusters of face selectivity. 1 cluster in the inferior occipital gyrus that I'm going to call IOG faces. Another cluster in the posterior part of the fusiform gyrus that I'm going to call P-fuse faces, and a third cluster that's about 1 centimeter to 1.5 centimeters more anteriorly that I'm going to call M-fuse faces. And a characteristic of these areas is that they respond more strongly to faces than to any other stimulus. And this response and the selectivity is maintained across formats. For example, if you show silhouettes of faces, they also produce higher responses than silhouettes of shapes or scrambled silhouettes. So it's quite an abstract response. We can also use electrical brain stimulation to test the involvement of these regions in perception. And occasionally we'll have patients, like epilepsy patients, that have electrodes implanted in their brain to test for the onset of epileptic seizures. And we had such a patient that we had electrodes implanted by chance over P-fuse and M-fuse, and you can use electrical stimulation to see what happens when you disrupt normal activity in this brain regions. And we did a series of trials, and I'm going to show you one of these trials.
All right, ready? 1, 2, 3.
Yeah, it metamorphosed again, and you look like someone I've seen before, but maybe a different person in my memory. Almost like your nose kind of shifted to the left a little bit, and your look just changed.
It did.
Yeah.
No.
Tell me more.
Well, I mean, I don't have a photographic memory, but it just, you turned into someone else. You looked like someone else.
Did I keep my gender?
Yeah. Oh, yeah.
How did you know I'm not a female?
Because you're still wearing a suit and tie.
Oh, you could see the suit and tie?
Yeah. Only your face changed. Everything else was the same. Yeah.
Did the skin color remain the same?
Yes. Yeah.
Did the position of my lips and nose and eyes stay the same when they got warped?
They shifted. Let's say they shifted to a side and maybe stretched, but they didn't get larger or smaller.
Okay.
It was more of a perception, how I perceived your face.
Interesting. Tell me more.
That's about all I can say. It just...
Okay, so using electrical brain stimulation like this really shows the causal involvement of these regions in face perception, because when you stimulate them, you get a specific deficit in the perception of faces. So now that we have a really good model system in the human brain to study face perception, how are we going to implement this in a neural network? So the network that Dan described before was inspired by the macaque brain, and usually it involves a hierarchy like this that goes from V1 to V2 to V4 to IT. However, in the human brain, you might notice that V3 is as big as V2, and in fact, bigger than V4. So one thing that when you want to model the human brain, perhaps you need to update the hierarchy, for example, include V3. Another thing that you might want to consider is that ventral temporal cortex is not a homogeneous piece of cortex, as it includes 3 stages of face processing, so you might also want to include these regions in your network. And finally, I do want to point out another difference between the humans and the macaques. Humans have an extra gyrus in the temporal lobe, and actually the gyrus, the fusiform gyrus, where face selective region is human-specific, and it doesn't exist in lower monkeys. So one thing that we might want to do is to generate a neurally accurate model of face perception is include a hierarchy that includes V1, V2, V3, V4, IG, P-fuse, and M-fuse faces. So not only that we need to update our hierarchy to generate a neurally accurate model of face recognition, but we might also want to consider how to compare stages in processing in the brain to stages in an artificial neural network. So one of the common approaches that people have used in the field is using something called representational similarity analysis, that you look at how representational spaces in the brain and in the deep neural networks might resemble each other. So typically what you do in a human experiment, you might put people in the scanner and show them different images, for example, an image of a hand and an image of a face, and measure the distributed response to these different stimuli across ventral temporal cortex, and then determine how similar these responses are. And typically, the metric is a correlation metric. And basically, each bin in this representational similarity matrix defines how similar two brain responses to different stimuli might look like. And then you might want to do the same thing in in an artificial neural network and see if these RSMs resemble each other. One of the things that came out of a large body of research actually in human neuroscience is that images of the same category tend to generate very similar brain responses. So you can see here this dark square means that the correlation between patterns of response to different face images is very high. However, if you look at the correlation between images of faces to, let's say, inanimate objects, they generate very different responses. So in this particular picture I took from a study by Nico Krieges-Korte, and what he did in the study is he generated RSMs in different stages of an artificial neural network and examined which processing stage resembles that of ventral temporal cortex in human. And what he found was that the higher stages of the network, kind of like Dan's work, basically the highest stage of the network generated the most similar representational structure as the human brain, and moreover, actually a linear combination, again, kind of like Dan's work, generates the best correlation. So this is kind of interesting because of two things. First of all, there is a tool for us to compare HDNNs to human brains, but it also suggests that if you have a network that in the highest stages has some sort of categorical responses, this might be a good network for the process of object recognition. So far, I've shown you a lot of similarities between the brain and HDNNs, but there are also some differences that I'd like to illustrate. So first of all, this representation similarity analysis is really interesting, but it's very abstracted from the way information is laid out in the brain. And if you're a person like me who stares at people's brain on a regular basis, you'll notice that the brain is actually organized in a very consistent manner across people. For example, if you look at maps of faces versus places or animate versus inanimate objects, they generate a very consistent topology or topography across ventral temporal cortex in which there is this lateral medial gradient. So it's like a representational axis of animacy is actually laid on a physical axis on somebody's brain. And furthermore, a large body of research from other labs as well illustrates that there are multiple maps, not only maps of category in ventral temporal cortex, but also maps of eccentricity bias and world object size that also have this lateral medial gradient. And this gradient is so consistent across people that if you identify a small sulcus in the fusiform gyrus that bisects it, it's called the MFS, you'll find that the MFS predicts the lateral medial transition in all of these four maps. So this is kind of interesting because it suggests that there's not only a functional structure coupling between the brain and macroanatomy, but it also generates a regular relationship across multiple maps. For example, lateral to the MFS, you'll find face representation, animate representation, foveal representation, and small objects. And medial to the MFS, you'll find place representation, inanimate objects, periphery, and large objects. And this means that there is this regularity among multiple representation laid out on the cortical surface. And another thing to note is that these representations might have different spatial scale. For example, the map of animacy spans several centimeters, encompassing the entire ventral temporal cortex. And you can see that within the animate compartment, you'll see more finer scale representation at the scale of centimeters of clusters that are preferring faces or bodies, and this is a second cluster of faces. And a lot of work from electrophysiology suggests that there is even finer scale, maybe at the scale of a cortical column, and at this cortical column of maybe 100 microns, you'll find clustering of neurons that might represent face parts or face viewpoint. So looking at the structure, we propose a hypothesis that there might be a relationship between the level of abstraction of representation and the spatial scale that's represented as the brain, whereby more abstract information such as distinction between animate and inanimate object is at the scale of several centimeters. Information about ecologically relevant categories such as bodies and faces might be at the scale of a centimeter. And at the level maybe of a cortical column, you might represent very fine-grained details such as features. And so this might be interesting, but it also might be good for something computationally in that it gives you a flexible level of information or a hierarchical information structure within ventral temporal cortex. And depending on task demands, you might read out different kinds of information by reading out information from ventral temporal cortex across different spatial scales. Now, this is now a difference between the brain and deep neural networks because the performance of the deep neural network doesn't really depend on how the neurons are organized within a layer. But this also gives an opportunity to test some hypotheses. For example, I've given a hypothesis that this spatial structure might give us some flexible readout of category information, and by comparing between different networks, one that has a spatial structure and one that does not have a spatial structure, it might give us an opportunity to test this kind of hypothesis. As was mentioned before, as the basic processing in the visual system is done by receptive fields. So what is a receptive field? A receptive field is a region in visual space that is processed by a neuron. And one feature of receptive fields in the human brain is that receptive fields that code similar location in the visual field are physically clustered. And this clustering is in the order of a millimeter in the human brain, which gives us an opportunity with fMRI. Since we cannot access single neurons, we can measure the visual field represented by the population of neurons in a voxel, and we call that a population receptive field, or PRFs. And the PRFs that I'm going to describe today involve a very simple mathematical model, and they include a two-dimensional Gaussian followed by a compressive nonlinearity. And again, this is a similarity between the architecture of HDNNs in the brain, because the basic operation that's performed today in these deep neural networks is a convolution operation with a filter, a spatial filter that operates on the image. And the hypothesized computational value of this kind of architecture is that it breaks out a difficult problem to a lot of local computations, and therefore it enables parallel processing of the image and increasing the speed of processing. A feature of the hierarchy of the ventral stream is that the receptive field size, and consequently the population receptive field size, increases as you ascend the ventral stream processing hierarchy. So for example, by the time that you're in phase selective regions, the mean PRF size is about 4 times as big as the PRF size in V1. And the same is true of the architecture of HDNN, because the pooling operation from a lower layer to an upper layer generates units in the higher stages of the network that see bigger and bigger portions of the image. So to give you some intuitive anchoring about how these population receptive field size might relate to your vision. So suppose you're looking at somebody standing in front of you, maybe he's there about a meter away, and you're looking at their face. What would a PRF in V1 see if maybe it's 2 degrees off the center? And you'll see that this PRF really looks at really local information, maybe something like the corner of your eye. By the time we ascend to V4, so a PRF in V4 might see a facial feature. Maybe it will see the eye, the eyebrow, and the corner of your nose. And by the time you reach to phase selective regions, PRFs in phase selective regions might include several facial features, or maybe even the whole face. So this suggests that they're kind of optimized to integrate across information across facial features, and that seems to be maybe the spatial basis for something like holistic processing in face recognition. So the idea is that this increase in the receptive field size across the processing hierarchy might generate more task-relevant receptive fields that are more useful for recognition in higher stages as the network. Yeah.
How do these receptive fields change with saccade? You move your eyes about, right? You become randomly in this.
So the receptive field doesn't change with saccade. Your fixation point changes with your saccade. So in this particular example, I showed an example where you're going to be fixating on the center of the face, shown here by the crosshair. And I'll show you a bit what happens under different tasks. OK, so this is what happens. So far I've shown you this hierarchy that's happening both in the neural networks and in the brain. But there are also some differences. For example, the basic architecture of HDNNs is that within a layer, you have the same filter that is involved across the entire image, meaning that the filter is fixed across a layer. Now, this is not true in the brain. So what happens in the brain in each processing stage, PRFs are actually differing in size depending on eccentricity. So close to your center of gaze in your fixation, the PRFs are small, and as you go from the center to the periphery, the PRFs or receptive field size changes. And this is true in every visual area, even as the average receptive field size increases across the processing hierarchy. Now, this is an example of a difference, again, between the brain and an HDNN. The question is it good for something? Or maybe this is a biological constraint. So what we think is happening in the brain, that the brain has a trade-off between resolution and limited, like, hardware. And the compromise that the brain has done is to reduce resolution, but reduce resolution specifically in the periphery while keeping the resolution high at the center of gaze. So again, these differences between the brain implementation and deep neural networks that might not have the same kind of physical limitation also can give rise for us to understand what might be constraints in biological systems. Another thing that might be interesting to look is how do these PRFs style the visual field. So in this example, I took all the PRFs in V1 in the left hemisphere and see how they cover up the visual field. And this is what we call the visual field coverage of a region. And as you can see, the left hemisphere covers pretty uniformly the right visual field. So in each hemisphere, we have a hemifield representation of the world. And this is typical of early and intermediate retinotopic areas. However, this is not true in face-selective regions. So now I'm showing you the, again, left hemisphere data, the PRFs in the face-selective regions. While we still have the centers being in the contralateral visual field, you see that the receptive fields are big. They always cover the fovea and extend to the ipsilateral visual field. And the consequence of that is that if we look at the visual field coverage, it's not uniform, but in fact we have much higher coverage of the fovea than the periphery of the visual field. And we call that the foveal bias. And work by Rafi Malach has suggested that this foveal bias might be related to the way that we look at faces. So you ask what happens when we do normal viewing of faces. So when people, in our case, we measured eye movements. When people were asked to recognize a face, they're shown an image that they may or may not seen several minutes before, and they have to say, do you recognize it or not? And we let people freely view the face. If you measure the density of fixations, you'll find out that many of the fixations, in fact, most of them, are close to the center of the face. This is illustrated in this plot. And the reason that we think that people are doing this kind of behavior because it puts their receptive fields in these face-selective areas in the region where there are informative faces for face recognition, and these are the internal features of the face. So this also just suggests another idea that maybe these PRFs in higher order areas might be task relevant. So to test this hypothesis, we measured PRFs across the ventral processing hierarchy across 2 different tasks. So in this task, we put people in an fMRI machine, asked them to fixate on a stream of rapidly presented digits shown centrally, and then we showed faces in 25 random locations. And as they're looking at these streams before each trial, they're giving a cue to what to attend. So they're asked to either attend to the centrally presented digits and report if 2 consecutive digits are identical, or they're asked to attend to the face without moving their eyes and report if 2 consecutive faces are the same person. And we ensure that they keep their eyes on fixation by measuring their eye movements during the fMRI scanner. And then we could test if PRFs are fixed across these 2 tasks, or maybe the task might affect the PRF properties. So if we look at face-selective regions, IOG, PFUs, and MFUs, and measure their PRFs under the 2 tasks, we see that attention affects PRFs. First of all, during the face task, you can see that the PRFs are more eccentric or more further from the center of gaze as compared to the digit task. So this suggests that attending to faces peripherally shifts the processing units towards the attended location. And notably, this property is specific to phase-selective regions because if we look at PRF eccentricity in early visual areas such as V1, V2, or V3, we really don't see a significant difference in their PRF location across task. Another interesting feature is not only that the PRF location changes, but also their size changes. So when people are asked to attend to a face compared to a digit, you can see it here, the PRF size actually doubles. So basically, there's a doubling in the PRF size during the face task compared to the digit task in all three face-selective regions. But again, in V1 or V2 or V3, we don't see a change in the PRF size. So this suggests that higher order areas seem to be modulated, their PRF seem to be modulated by task demands, whereas early visual areas are not. So what are the consequences of this change in the PRF allocation across both tasks? One can think about understanding this by looking at the visual field coverage under two tasks. So this is the left diffuse faces. These are all the PRFs in this region under the digit task, and these are the same PRFs under the face task. And you can see that during the face task, the receptive fields are bigger and more scattered, and consequently extend much more to the periphery. So that means that if you have a face in the periphery, it's covered by more PRFs during the face task compared to the digit task. And this might have some computational consequences. For example, maybe because of this difference in visual field coverage, you can better detect a face in the periphery in the face task compared to the digit task. And in order to test this hypothesis, we did what's called a model-based decoding. We took the responses of the PRFs under the digit task and then under the face task and estimated how well they can dissociate the location of the faces in different parts of the visual field. So this example shows the spatial error in identifying the location of faces in the right upper quadrant. And you can see that there's a lot of error during the digit task, but much reduced error during the face task. And in fact, we can quantitatively measure that, and there's a fourfold reduction in the spatial error during the face task than the digit task. So this again suggests that we have PRFs in the human visual systems that might be modulated by task and give us task-optimized processing. And again, this is a big difference between the brain, and maybe the brain can use these kind of top-down connections like Dan had mentioned in the brain to provide for task-optimized processing. Sometimes insights actually come from the computational literature rather than the neuroscience. And I think that one of the most profound insights from the computational work was that learning is key for generating good filters for object recognition. For example, this is a slide from Krzyzewski and Hinton's seminal work in 2012, in which they trained a network to categorize all the images in ImageNet. And then they looked at the features in different layers of the network, and they found that in their artificial neural network, the features in the first layer resemble that of V1. And the idea is that these filters might be optimized for extracting the natural image statistics across a broad range of object categories. So this, of course, also leads to an obvious question, whether PRFs in the human visual system are also amenable to experience and maybe experienced during human development. So what we did in our experiments, we wanted to test if PRFs are changing across childhood and adulthood. So I'm going to describe to you experiments that we did in 25 children and 25 adults. We used children between the ages of 5 and 12. Before age of 5, we cannot really get children to be still and attentive in our scanner. So it's going to be very difficult to do these experiments in infants, as Dan would like us to do. And nonetheless, this is an age of a lot of development in the child brain, as there's a lot of improvement in face recognition and reading that involves these higher order areas. So what we do in this experiment, we have participants lie in a fMRI scanner. We have them stare at a central stimulus that's a little bit more child-friendly. And when the stimulus changes color, the participants press a button. And as they're attending to the center, we have a high contrast bar with checkerboards sweeping across the visual field. And again, we make sure that the participants fixate, and our children can easily fixate across this target. And what we did is we measured PRF properties across the ventral visual processing stream between children and adults. So this is the average PRF size from across the ventral visual hierarchy in children in light colors and adults in dark color. So you can see that children, like adults, have increasing receptive field size as you ascend the visual hierarchy, and in fact there is no difference, quantitative difference in the PRF size across children and adults. If you look at the visual field coverage across, again, retinotopic region and the ventral stream, in both children and adults, you see this really nice hemifield representation, and there seems to be no significant difference between children and adults, at least in retinotopic regions, in the ways that the receptive fields tie as a visual field. So at least by the age of 5, the early retinotopic areas seem to be fully developed. So what happens in higher order areas? So this is a visual field coverage in the child right P-fused faces. And you can see that in the right hemisphere, now it covers the left side of the visual field, and you can see this foveal bias. As you can see, there's a higher coverage in the center of gaze. But you can also see that a lot of the coverage is actually on the lower left visual quadrant. But this is a different visual field coverage than what we see in adults. As you can see that there are two changes that are happening. First of all, the foveal bias seems to be increasing from childhood to adulthood. As you can see, there's a higher density of foveal coverage in adults. And we can quantify this by measuring the center of mass of the visual field coverage across children and adults. And you can see that in the right hemisphere specifically, this foveal bias increases from childhood to adulthood. Another thing that you might notice is that the area of the visual field that is covered by the adults' PRFs is bigger than in the children's PRF because it extends into the upper visual field as well as the ipsilateral visual field. So, and in fact, if we measure this, this is about double the visual field coverage in adults as compared to children. So we see in these higher order phase selective regions a profound development of PRFs after the age of 5. Yes? 5 minutes, okay. So what does this mean in terms of your behavior? So if you're a child and you're presented with the following stimulus, let's see where your visual field coverage happens in your phase selective region. So I'm just plopping it the right P-fused faces visual field coverage, and you can see that most of the places where you have the highest coverage is actually outside the region where you have interesting features for face recognition. So what you should do as a child is you should move your gaze upwards and rightwards because that would put your visual field coverage on the place with informative features. And we could test this hypothesis by looking at where children and adults look at faces when they're asked to do a face recognition task. So in this example stimulus, this is the adult's fixation, and this is a child fixation. And consistent with our prediction, you can see that the child fixation is shifted rightwards and upwards, whereas the adult fixation is on the center of the face. And we can do this from many images, and we can see the bias in the child's fixation compared to adults using a vector. And you can see that there's a significant bias that childs fixate upwards and rightwards compared to adults. And basically, they never fixate downwards and leftwards because that would push their visual field coverage actually even more outside of the face. So this is kind of interesting because it shows that experience still molds our PRFs even through our childhood development, and it also suggests that this is related to understanding where our informative features in the faces. So again, the idea that in the brain as well in the HDNNs, the filters are learned is probably useful because it lets us adapt to our natural images statistics and also adapt our processors for particular tasks that are important for us on a daily basis. So to wrap up, I hope I've convinced you that this is an exciting point in the field, that we can combine advances in both HDNNs and neuroscience to build what I call neurally accurate models of the human ventral stream. But in order to make HDNNs neurally accurate, we also have to make changes to the present architectures that are prevalent in computer science. Specifically, we might want to consider how to implement HDNN that actually have spatial topologies. We need to think about what's going to change in the HDNN if we're going to have filters that change with eccentricities. We might need to think about implementing filters that are task adjustable and not only during the learning stage. And I haven't talked about it, but there's also a lot of temporal properties to these filters. And as Dan has indicated, right now, most of the architecture is static. And my great hope is that this computational understanding will not only lead to generative models of the brain, letting us to better be able to predict brain responses, but also provide important insights about the computational utility of specific implementation features in the human brain. So thank you for your attention.